Hello everyone, welcome back. This is Lab 12, Aquatic Species Diversity. So just to let you know, as you've probably seen before on the D2L, all of our labs and our pre-labs will be completed as quizzes online. I know this is unorthodox and I'd rather be taking you outside for labs like these, but of course during these times we don't really have much of a choice in the matter, so we'll do the best we can with what we have. So, today's lab we're looking at biodiversity, different life forms or species and life-sustaining processes that can be best or that can best survive life on Earth. Generally, when we think about biodiversity, we think about different types of species, and that is one type of biodiversity. But there are four different kinds in general. We also have we have genetic diversity, we have species diversity, ecological diversity, and functional diversity. Now, genetic diversity is variety in genetic makeup of a species. And for example, if we look at a group of humans, for example, we can see that, and this is of course if we had a class of students, I could show you this, but no two individual humans are exactly alike. The vast majority of us share a great amount of our genetic code, but of course you can see that there are differences between hair color and skin color and eye color and other observable features like height. And that tells us that there are genes that differ amongst each one of us. In, in general, it's about 0.5% of our genetic code that differs from one human to another. And we can also see it in wild organisms like this herd of horses that no two individuals are exactly alike. And that's actually important in conservation because when you have two organisms that differ, that means that it can actually promote the survival of a species. If no two individuals are alike, you have better odds that some of these organisms will survive if you have dramatic environmental change. Um, Whereas if organisms are genetically identical, there are higher odds that all of them will be wiped out if you have some sort of natural disaster or something like that, like a wildfire or um, a disease or something like that, especially. We have species diversity, variety among species and habitats. So you have two different examples of here. In this example down here, you have the Amazon rainforest with over 150,000 different species of plants and animals. And on the right, you have the Arctic, which only has about 1,700 species. And the difference between the two is that the habitat for the rainforest, it's um, much more conducive to life, with warm temperatures. There's plenty of food choices to access. And so you have a wide variety of species that have adapted to these different food sources and to exploit them, essentially. So it produces a great diversity in this rainforest. On the other hand, the tundra, it's a much harder place to live. It's less forgiving. And there's very few food sources to choose from. So most organisms have adapted just to survive these harsh conditions. And so there's much less diversity overall. So the type of environment, the climate, the food sources can all dictate how diverse these environments are in terms of number of species. We also have ecological diversity, variety of habitats such as forests, grasslands, and streams. So for these types of diversity, what we're looking at is just the number of different habitats you have in a given geographic area. And this can actually influence the biodiversity we talked about earlier, biological diversity, when you have different habitats in one area. So for Colorado, for example, you might have this mountainous or alpine habitat at high elevations. You have forests consisting mostly of conifer trees. You have open prairies, meadows, and you even have streams, and of course with each or lakes. And because you have such variety in habitats, it means that there are likely to be different types of organisms living in each habitat. And of course, like I said, that can mean you have a higher biodiversity, so or species diversity. So in this case, a wide variety of habitats lends to a higher number of species, different species. We also have functional diversity, the physical or chemical functions of the organisms in an environment. So here we have two environments side by side. And In terms of number of species, they both have the same number. If you look at the left, you have one, two, um, three, four different types of species here. And those consist mostly of, in this case, three types of barnacles and one type of mussel. And on the right, you have four species. You have seagrass, you have anemones, you have barnacles, and you have starfish. So both ecosystems have four different species. The big difference is between the two is that these are mostly the same types of organism, like I said, barnacles, whereas there's a much greater diversity in types of species here. And the big difference is the function of one of the species in this ecosystem, and it's the starfish in particular. You may remember from lecture that starfish play a very important role in eating shellfish like mussels, and they act as a very important species, this predator, because what they do is they promote diversity by their presence and activity of feeding on mussels. The mussels would normally outcompete other organisms, likewise with barnacles. So if you have a starfish, a major predator, it's very likely that it will keep those numbers back and allow other species to thrive, like these anemones and this seagrass here. So this one species plays a very important function in its ecosystem, and when you lack it, the ecosystem as a whole kind of suffers in terms of its variety. Now, human actions, including agriculture, elimination of species, pollution, deforestation, and human settlement, can all have effects on biodiversity. Um, a lot of these activities, like for example pollution, can result in losses of biodiversity. A good example here is eutrophication. Eutrophication, if you don't know from lecture already, is a situation where you have too much, too many nutrients in the water, like fertilizers, like nitrates and phosphates in the water, and what that does is it causes a bloom of aquatic algae. And when that happens, the algae overpopulate and they suck all the oxygen out of the water, and you get a massive die-off of fish as a result of that. And of course, then you may have a chain reaction where if the fish die, the organisms that eat them may also die, and so on and so forth. And of course, you would see that that would have a huge effect on biodiversity in the ecosystem. And if you're not aware, of course, if we're talking about fertilizers, you might think of like an agricultural area, but this is a residential neighborhood with fertilizers coming off of lawns, for example. So a lot of people don't think about how that may impact local lakes and waterways. Loss of biodiversity also has known and unforeseen consequences and often increases the vulnerability of an ecosystem to further disturbances. A good example of how we affect biodiversity, and in fact, usually we have no idea how we've affected it until it's a little bit too late, are honeybees. They're important pollinators of the flowers of North America. Since 1947, they've suffered a 60% reduction in number. And this is important to us for two reasons. First of all, because we depend on these honeybees, especially the European variety, which we've domesticated because they actually pollinate a lot of our crops. But when you think about it in terms of their ecological role, a lot of them pollinate many native flowers, many native plants. And so without these honeybees, these plants cannot reproduce. And so we see an overall loss of species. Another chain reaction that occurs there is when you have the pollinator disappear, the plants that depend on it disappear. This lab focuses on species diversity, the proportion of different species within a community. And it can be measured in two ways. We have species richness, the number of different species, that's just where you count the number of individual species, and species evenness, the relative proportion of each species. So we have two communities here. We have community 1 and community 2. If you look at both of these two communities, they have the same richness. They both have 4 different types of species, A, B, C, D in community 1, and then in community 2, the same species, A, B, C, and then D. Okay, so the same richness between the two ecosystems. However, the big difference between the two, as you've probably noticed, is that one species is particularly abundant in community 2. So if you look at the relative proportion of each species, that's where we look at evenness. In community 1, each group has the same number of individuals. There's 5 individuals for each species. And so that means that each species occupies about 25% of that community. On the other hand, community 2, you have species A dominating the ecosystem. Makes up the vast majority of individuals in this ecosystem. In this case, 80%. So that being the case, even though they both have the same richness, there is a huge difference in the evenness. and it can actually produce an illusion, um, in this case, that the ecosystem is not quite as diverse as it really is. In addition to that, when we see a species that dominates in one particular community, that could tell us something's wrong with the community. In this case, maybe that this species is dominating because of maybe climate, for example, like climate change. Maybe it does well when it's too hot or too cold for other species. Maybe the other ones are too sensitive. It could also be an indicator of maybe pollution, whereas this species A could be resistant to pollution in the soil, whereas the other species are more sensitive to it as well. So it could tell us that there's something wrong with the health of the community when you have something like that happen. Now, some species are disturbance adapted, which means that they do better than other species when a habitat is facing major change, such as natural disaster or human activity. Again, as I mentioned here earlier, maybe species A could do well with environmental change. You may remember that a disturbance is anything natural that could happen, like a fire or a flood, like a wildfire flood. Maybe even a hurricane or a tornado sweeps through an area and decimates it. Or you could even have human activity like clear cutting or global warming. But some species are just going to do better than others. They're what we call disturbance adapted. A good example, and you'll see this in your textbook, is that we have a species called the northern spotted owl that lives in Northern California. It lives in these old growth forests of these old growth continuous forests in Northern California. Certain species like the northern spotted owl depend on these vast tracts of forest because they need to nest in old growth trees, and in addition to that, their primary food sources also thrive in these vast forests. The problem is that these forests are now disturbed. Clear cutting has fragmented all of these forests into patchy areas, and this means that this cannot support these owls. They're losing the old trees they depend on, and also, of course, their food sources. And in addition to that, we've also thrown an invasive species into the mix. So on the left, you have the barred owl, which is from eastern North America, and on the right, you have your northern spotted owl over here. They're very similar species, which means that when you introduce them together, they compete with each other directly. And the barred owl, unfortunately, is a disturbance adapted species, so it does well in these fragmented forest habitats. Of course, the northern spotted owl does not. And we can see that this is having an overall effect on the community because the barred owls are actually increasing in number. There's 2,000 recorded individuals in Northern California, where there's 560 pairs of the spotted owl in Northern California, so their numbers are dropping. Okay, so like I says, in the fragmented forest in Northern California, barred owls are thriving, with their numbers increasing, whereas the northern spotted owls have fewer places to breed and hunt, so they are on the decline. So hopefully you should have the impression by now that biodiversity can be an indicator on the health of an ecosystem or community, whether you have too few species or whether you have one species that becomes particularly abundant. It can be an indicator that something is wrong with that ecosystem. And in this lab, we're going to be looking at aquatic communities, in particular a pond. And so measuring biodiversity in an ecosystem like that could actually give us an idea of the health of that aquatic ecosystem, maybe the water quality of the ecosystem, like, for example, telling us whether it's polluted or not. So how do we measure biodiversity? Well, in this lab, we actually use what's called the sequential comparison index. It's a simple method used to evaluate biodiversity, primarily in aquatic communities. It is a number between 0 and 1. And as you'll see, I'll show you how you measure this in just a minute. But looking at this number, if you get a calculation of above 0.6, this indicates a healthy aquatic community. It means the water quality is fairly good because you have a diverse number of species in that ecosystem. A low index below 0.3 would indicate poor water quality, as only a few species would survive in this polluted water. Now, looking in this lab, we look at macroinvertebrates. A macroinvertebrate, first of all, an invertebrate is an organism without a backbone. But a macroinvertebrate is one that is large enough to see with the naked eye. Most of the organisms that are invertebrates are microbes, and so we cannot see them. But if it's large enough to see without a microscope, we call it a macroinvertebrate. So we're talking about things like crayfish, aquatic beetle larvae, freshwater worms, leeches, for example, snails, all fall into that category. And macroinvertebrate samples are collected with a dip net, and this is what we would use in lab. and then sorted by hands or by tweezers using trays like we see below. So here's an example of different types of macroinvertebrates. And you don't have to know any of these by name or ID them as such, but if you are curious about what macroinvertebrates are out there, there's a link down here at the bottom of the PowerPoint. Now, when determining SCI for aquatic organisms, they are sorted into rows on a tray. You begin counting from left to right. Now, first of all, in lab, if we were doing this, it'd be very difficult to organize your species in rows, so we do the best we can because they're alive, they're going to be moving around, but we do the best we can to organize them. And then you're going to put them in, like I said, put them in rows, and then you count them from left to right. You score the number of adjacent organisms that are the same species. Okay, so they have to be identical species. And for each row that consists of, or each sequence that consists of individual similar species, we would count that as a run. And you continue down the row counting your number of runs, your number of groups of individuals, groups of similar species, and then you continue on to the next row as if there is no break. So basically you're simulating that it's just one line of species. And each type of organism is denoted by a letter. Again, like I said, we're not identifying what these organisms are. What you can do is just recognize that they're different types of species, that they won't look the same. And so instead, you just denote each new species as a different letter. So let's look at an example here, just easier to show you. So here we have a sample of macroinvertebrates taken from the water. And what we've done, just assume this white space is a tray. And we're organizing these species into this or across this tray and into rows as best we can. So our first organism is a dragonfly larvae here, which we will designate as letter A. Okay? And so here we have two dragonfly larvae, and we've indicated them as letter A here. And so we're going down the sequence, and we hit a new species, so we designate that as B. And we keep going. We hit another new species, C. And here's our dragonfly larvae again. Okay? So remember I said that we're assuming this is one line, even though we've kind of moved it into multiple rows. So as we move to the next line, we have the same species. So we still designate it as A. There's another one. So that actually counts as one run, as we'll see. Okay, so they're all the same species. Then we have B. Okay, we've seen that one before already. Then we have a new one, so we'll designate that one as D. And then we have another one that's new, E. We have another new one, F. And then finally, there is another one we've seen before, that is D. Okay. So then what we do is we count up the number of runs. And so remember, those are the number of organisms, adjacent organisms that are the same species. Okay. So we have two A's side by side. That is one run. Okay, we have B. Okay, so we have a new species, so that's a new run. Then we have C, another new run. So we're back to our dragonfly nymph or larvae, that's A. And then we continue that on to the next row as if it's one run. Okay, so that's another new run. Okay, B, another new run. D, another new run. New species here, another one. And then another new run here. And then finally we have our last. Okay, so again, like I said, what we're doing is we're counting the number of adjacent individuals that are the same species as a run. So we have 1, 2, 3, 4, continuing on to the next row, 5, 6, 7, 8, 9. And to calculate SCI, what we do is we divide the number of our runs by our number of individuals. So we have 9 runs, and we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 species, or excuse me, individuals on this page. And so to calculate SCI, we divide the number of runs by number of individuals. And so that's 9 divided by 12, and that gives us 0.75. And if you remember, I said that when we calculate SCI, if it's a number above 0.6, that indicates a healthy aquatic habitat. If it was below 0.3, it would be considered, or in this case, it would be considered poor. Something would be wrong with that ecosystem. So here you have an example that I want you to calculate and work through on your own. You're going to be calculating SCI for this aquatic habitat. Okay, so answer questions 3 and 4 on your lab exercise here. So let's imagine we're sampling the aquatic community at the pond Lake Teal, located on the College of Coastal Georgia campus. We select 4 sites to sample around the perimeter of the pond. Okay. So this is a pond that is in a heavily populated area with a lot of human activity, and we would take our students out here and we would sample using those dip nets I told you before and calculate, or in this case, count the number of different individuals as well as the ruins in each one of these 4 areas. And here's where we would be sampling in this exercise. So 4 sites. And for each site, these are the SCIs we get: 0.31, 0.39, 0.28, and 0.17. So for this area, what I want you to do is to calculate the average of these SCIs for Lake Teal. And then you're going to determine, well, what is the overall health of this aquatic community? Now we're going to sample a different location, far away from the main campus, and a bit more secluded, Horton Pond, in Jekyll Island, Georgia. So this is a much more remote, more secluded area with less people interacting with it. And we're also going to survey 4 sites on Horton Pond. And so for this particular area, we get 4 SCIs: 0.78, 0.67, 0.81, and 0.77. And you're going to do the same thing here. Calculate the average of your 4 SCIs, your indexes, and then you're going to determine the overall health of this aquatic community. And so lastly, I want you to kind of compare the two different. I mean, of course, you've got the 2 SCIs, you've got the health that you've kind of determined for your two communities here. And I want you to kind of explain to me in the last question, what could account for the differences that we have between these 2 ecosystems? Why is the SCI, the overall SCI, calculated so much different between the 2 communities? What could you interpret from this? All right. So that is it for this week's lab video, and the handouts. I mean, there is a handout for this online, but the exercises, again, like I said, will be due by the end of the week on Friday at 11:59 p.m. All right. Everybody have a good weekend.